So that was a fantastic segue to my talk, whoever asked that last question, because I'm going to actually tell you what happens to mRNA after it's made by on DNA. There's many steps of gene expression. There's, of course, transcription that many of us know and love. And then there is the whole life cycle of RNA, where RNA is made on chromatin. It's exported from the nucleus, finds the ribosomes, and then eventually degraded. And each of these levels of gene expression can be regulated in order to ensure that we get the proper assembly of protein complexes. And the reason why me and others study all of these layers of gene expression is to understand how subunits of different protein complexes are made in nearly stoichiometric ratios, because if you make too much of a particular protein, you can overwhelm proteostasis pathways, which can lead to disease and aging phenotypes. So a number of years ago, a fantastic student in my group, Brendan Smalek, teamed up with a wonderful computational biologist in my group, Robert Eastwart, to monitor RNA flow throughout the cell. And the idea there was that if we could track RNAs as they move throughout their life cycle and the cell, we could identify sort of bottlenecks in their trajectories. And presumably these are places where regulatory control is applied. And so they set out to answer this question: what are the kinetic rates of RNA transit across the cell, and to measure these rates really precisely. And so we decided to use metabolic labeling because it is a really classic approach for following RNA life cycles. The idea is you use a uridine analog and that's labeled, and you add this to the cell, and RNA polymerase incorporates this analog into the growing RNA. And then as a function of time, those RNAs proceed throughout their life cycles. And if you purify the RNA, you can detect which ones have the label. You can calculate the percentage that have the label or are newly synthesized since you added the label. And from that, you can calculate half-lives for each RNA across the cell. And what Brendan wanted to do was, instead of looking at the total half-life of the RNA, was to couple this approach with an RNA, with cellular fractionation, to look not only at whole cell half-lives, but also this half-life that the RNA has in the cytoplasm and the half-life it has in the nucleus, as a way to identify how long RNAs are spending in different compartments across the cell. And this is actually a very classic approach that we took inspiration from, where decades ago, radio-labeled nucleotides could also identify newly synthesized RNAs. And then cellular fractionation, as you're seeing here, could show us that RNAs accumulate in the nucleoplasm, then later in the cytoplasm, and then finally on polysomes. Of course, when you use radioactivity, you're looking at all of the RNA at the same time in bulk, and you don't understand how different RNAs are transiting throughout their life cycles. And so we decided to take modern approaches in order to get at these questions, and we are using 4-thiouridine, which is a uridine analog. You add it to cells, it's incorporated into RNA, and then we can use biochemical fractionation into nuclei, chromatin, cytoplasm, and polysomes in order to purify the RNAs from these different cellular compartments. And then we use nucleotide conversion chemistry, which basically adds a bulky adduct to the 4-thiouridine that, upon reverse transcription, converts that uridine into a G, and then when you sequence and align it, you get T to C conversions that you can identify bioinformatically. And this is actually the hard part, the bioinformatics. Right here, I'm showing you the chemistry that allows the conversion during reverse transcription, and then you identify them bioinformatically. So this is actually the tricky part, and I'm going to gloss over it. But if you're interested in how this is achieved, I encourage you to explore these publications and our own. But at the end of the day, what you get per gene is an estimate for the fraction of new RNA as a function of the 4SU pulse. And so you can see that here for the gene TFAM, where the color here is indicating the number of mismatches, T to C mismatches per read. And you can see that it increases as the labeling time increases. And that can then be converted into a fraction of new RNA. And it can also, then you can look at this completely as a function of time. And what we're looking at here now is MYC as an example gene, and we're looking at the RNA that was purified from chromatin as a function of 4SU pulse. And you can see that by about 1 minute, 100%, or sorry, 60 minutes, 100% of the RNA now has the label. And so 100% of the RNA is newly synthesized since we added the label. And so this is for the chromatin fraction, but we also have the nuclear fraction, the cytoplasmic fraction, and the polysome fraction. And you can see that it is taking time for RNA to be released from chromatin, then leave the nucleus, and then find the polysome. And we have this also for total RNA as sort of a reality check that the other compartments should add up to the total RNA picture. And then we have it, of course, for all genes. This is for NIH3T3 cells. So this is a mouse cell line. And here you can see 3 example genes. But if you look at all of the genes together, you can see sort of a global picture, much like you would have seen by radioactivity. So here is the chromatin fraction of new RNA as a function of time. And the bar plots here are showing you all of the genes across that we've measured. And then the nucleus, you can see that the fraction of new RNA is coming up a little slower, cytoplasm even slower, and the polysomes slower still. And this really mirrors exactly what was seen decades ago by the Penman group, where they used radiolabeled precursors. But with our data, we wanted to go a lot further and actually determine the kinetics of RNA flow and use modeling in order to estimate the rates of turnover as the RNA is moving throughout the cell. And so using ordinary differential equations, this has been done before to model the whole cell turnover, as I've indicated here. And so if with a estimate of the new RNA, you can actually measure the whole cell turnover. And so we can then do that with lots of compartments, and I'm not going to write down all of the equations, but basically we modeled it as follows. We took a very textbook-based model where RNA is made on chromatin, it's released from chromatin into the nucleoplasm at some rate, and it's exported at another rate. It's then accumulates in the cytoplasm. At some rate, it finds polysomes, and then at another rate, it's degraded. And so this leads to a lot of rates and a lot of populations and a number of differential equations that can be solved for using our data. And so using a Bayesian approach, we were able to fit these data using those ordinary differential equations. And that's what you're looking at here, along with the confidence limits. And that worked really well. This textbook model of the RNA life cycle worked quite well for most genes, as you can see here for MYC and FOXG1. However, there was a really interesting set of genes that were outliers. For example, RPS24, a really important ribosomal protein gene, where, I should say, we only fit the models for all of the data set except for the total RNA. We use the total RNA as a reality check. Like I said, at the end of the day, the compartments should add up to the total RNA picture. And you can see, and so we predict the total RNA data from the results of the other, from the results of our models. And you can see that it worked quite well for MYC and FOXG1. We have a nice, estimate of the total RNA, but it really failed for RPS24, where we're predicting that the total RNA is much below, and that should turn over a lot slower than what we actually measure. And so when we saw this for this gene and many others, we realized, okay, it's looking like total RNA turnover is faster than cytoplasmic turnover, and it looks closer to the chromatin and nuclear data. And we thought that one way that this could be occurring is if there was a lot of nuclear degradation of the transcripts encoded by this gene. And so we went back to our model and we added in a nuclear degradation term. So now we have an extra rate where RNAs can be exported or they can be degraded. And we then use this new model. And indeed, in this case, we found that, yeah, we are doing better. We're predicting the total RNA data a lot better with nuclear degradation. And then to compare the two models, we can calculate a Bayes factor that discriminates between the two models and takes into consideration the fact that there's an extra parameter with the nuclear degradation and calculates the probability that each model is the better fit. And then we can actually plot something called the Bayes factor as a function of nuclear degradation rate. And the cutoff here normally in the field is about a Bayes factor of about 100, which means that the alternative model is 100 times more likely than the null model. And we indicate that these factors here are candidates for experiencing large-scale nuclear degradation. And when I say large scale, I do mean large scale. About at least 65% of the transcripts arising from these genes never make it out of the nucleus, and they're degraded there. And what we noticed is that, as you can see here in the yellow, and we call these predicted to undergo nuclear degradation, or PUNs. And these are genes where the data is best explained with the nuclear degradation parameter. When we look at who these genes are, we found that RNA splicing factors were really enriched in these genes. And this is something that we're really interested in following up on, trying to understand how these genes are targeted for nuclear degradation and what the role of nuclear degradation might be in terms of regulating these genes. But I don't have anything that I can show you today on that, but please stay tuned and hopefully we'll have something in the future to tell you a little bit more about these really interesting genes. But I'd like to return to sort of the canonical genes. What did we learn about all of the genes that are producing RNAs that persist through their life cycle in this more textbook manner? And so the first thing we can do is just look at the half-lives of each of these RNAs in all of the different compartments that we measured. And so what we can see here is just violin plots for the half-lives as a function of compartment that's indicated down here. And you can see that there's a number of take-homes from this. First of all, it's been appreciated for a while that RNA turnover globally is highly heterogeneous. And we, of course, recapitulate that with our studies. As you can see here, that the lifespan of different RNAs varies across the genome. But we saw that the rest of the rates as well vary just as much. And we call these RNA flow rates to indicate rates of RNA transit across the cell. The second thing we noted is that chromatin and nuclear half-lives are very similar. And this is really telling us that once RNA comes off of chromatin, it doesn't persist for very long in the nucleoplasm, and it will be exported quite quickly, as you can see here by our measured export times. Also, interestingly, is that cytoplasmic half-lives were longer than nuclear half-lives, but not that much longer. And I think that's really important to take into consideration when looking at RNA abundances and comparing abundances across the transcriptome, is thinking about how those genes are actually spending a lot of time in the nucleus. And actually, there's a lot of genes where their RNAs are spending a majority of the time in the nucleus. And so these differences in abundances that we look at and think about, it's more complicated when you think about the fact that they're spending their life cycles in wildly different ways across the genome. And finally, we could measure an estimate for polysome loading, and we found that it's fast, but it's not instantaneous. It's not as if the RNAs emerge into the cytoplasm and immediately find a polysome. So with all of this heterogeneity we wondered why. And one of the first things we thought was that maybe, genes of similar functions might have similar life cycles or flow rates throughout the cell. And that would be indicative of the fact that they are co-regulated at many steps across their life cycles in order for them to in order for their gene products to perform the same function at the right time in the right place. And so what we did is we clustered all of the genes by all of the different rates that we measured and all the different replicates, et cetera. And we got a number of different clusters. And in almost all of these clusters, we found many enriched Go terms. So for example, this cluster here is enriched for the cytosolic ribosome. Here we have genes that are involved in nucleosome assembly. And down here, we found a lot of genes that are involved in our transcription factors or involved in transcription factors. And so really, it looks like if you're functionally, that genes that have similar functions in the cell are co-regulated throughout their life cycles. So again, with all this heterogeneity, we also wondered, what's the molecular determinants of RNA flow? What might be controlling this? And to address that, we turn to machine learning because, as you can imagine, there's a lot of possible determinants. And basically, because we did one of our measurements in K562 cells, human cell line that was one of the main ENCODE cell lines where there's a ton of data, we decided to just write down all of the possible ways, all of the different factors that we thought might be impacting RNA flow. And as many can appreciate, of course, gene length we imagined would impact RNA flow because you're going to be on chromatin longer if you have a longer gene. We put in transcription factor binding, histone modifications, and then for RNA, we put in 5' UTRs, the length and the sequences and the motifs. We put in RNA modifications, intron lengths, microRNA binding site, poly A tail lengths, et cetera, and most importantly, RNA binding proteins, which was a really nice addition of some of the latest ENCODE projects that we could also interface with these data. And then we used a lasso model to identify which features were most predictive of RNA flow at a large scale. So this is a model that allows you to identify features that can predict what you've measured for a large amount of the data. And here are some of the things that we got out of that model and that were shown as important. We saw gene length, unsurprisingly, was important for the amount of time that RNAs spend on chromatin. Same with intron length, the number of exons. We saw a relationship between poly A tail length and persistence time on chromatin. And we saw a relationship between a variety of RNA binding proteins, as well as microRNA sequences. And so we followed up on a few of these. We found that DDX3X was indeed controlling the export of its targets. And we were able to verify this independently through knocking down DDX3X and through a CRISPR screen that looked for factors that rescue a DDX3X knockout. And we found many, many factors involved in export. In fact, it was the most enriched pathway of that CRISPR screen. We also found that PAB-PC4 controls the export of its targets to some degree. And by measuring the poly A tail length by nanopore sequencing across these cellular compartments, we could confirm that tail length correlates strongly with residence time on chromatin. So just the take-homes of what I showed you today. RNA flow rates are highly variable genome-wide. Like I said, really important to consider this when comparing different transcripts, and any perturbation is that you could be altering the abundances, but you also could be altering how long RNAs are spending in different compartments. All of the stuff that we measure at the whole cell level, of course, is a convolution of these rich RNA life cycles. And I just showed you, a simple lasso model that pulled out many different features of interest. Of course, There's more from that analysis than when we followed up on. And there's more sophisticated analyses where we could actually go, what we'd like to do next is go into cluster by cluster and identify factors that are critical for each of those clusters of RNA flow rates to identify sort of sub-mechanisms of all of this. And with that, I will thank my lab. I already thanked them, Brendan and Robert, who are now alumni who spearheaded this work. They had a lot of help from Eric McShane and Karen Choquet and other members of the current Churchman lab. And I'd be happy to take your questions.
We have time for a couple of quick questions. Winston, you can get to go first.
Okay. Hi, Stirling. Great talk. So my question is, you talked a little bit about splicing factors in the nucleus, but like as a class, what's going on with non-coding RNAs? Do they behave in the same way? Because you're talking a lot about coding with the polytome loading and everything like that. And as a quick sort of follow-up or opening it up for you, have you looked at all at non-polyadenylated RNAs?
Yeah, it's a great question and something I get asked a lot about. We just looked at coding RNAs because the textbook models are so, clear and it's straightforward to model those RNAs. And we haven't looked at non-coding RNAs yet, although of course they are in the data set. Mm-hmm. But the other problem with, maybe not all non-coding RNAs, but some of them are very short. Right. And so anything that's too short, we won't identify enough conversion events in order to quantify properly. But I agree that there's so much in this data that we haven't even scratched the surface of analyzing it fully. So I hope that it serves, we're following up on some of it, but we hope that also the community really enjoys it as a resource and as a great way for building hypotheses.
Awesome, thank you.
Hi, great talk. I wished you were here so we could talk to you a lot more about this. So my question is regarding the splicing observation that you had. How do you differentiate between degradation versus splicing or any RNA that has a processing workflow? How do you differentiate between that? And if you have any insights on that, looking at it more closely?
Yeah, I think it's a great question. So far, we're just looking at exons. And so we're not counting the intronic signal, but actually I think it would be very interesting to look at the intronic signal. And that's something that we'd like to do in the future. Haven't dug into it quite yet. But it's, so we just sort of ignore all of the transcripts, intronic sequences. And so it just doesn't even come into our models.
Yeah, I think this would be a great tool for measuring splice rates, right?
I would think that my lab would have done that by now. we look at splicing in other ways, but it's on a long list of things to do with these data. Thank you. And hopefully next year I can come. I would love
one more question. Hello, Stirling. Awesome talk. I'm wondering, I know you mentioned that there's so much more to mine from this data set, but I'm wondering about ER-associated transcripts and if they have a vastly different cytoplasmic half-life, and if there's earlier determinants or the early stages in the RNA life cycle are different between ER-associated versus non-ER-associated transcripts.
Right. Yeah. We didn't see, we looked at that early on and I can't remember what we decided. It's hard to, what we would like to do is actually label RNAs using APEX or something like that localize to the RNA and really look at their arrival there and do that timing as well. One of the caveats to this whole approach that I should, is that when you do a nuclear fractionation, you do end up getting, obviously I made it sound like all of our fractionation was very clean. It's not 100% clean, and the nuclear part does get enriched for ER-associated transcripts. And so I think when we started down that road of pulling out transcripts that were ER-associated versus, not, we were a little worried that if we that perhaps that all of a sudden that background might be complicating the picture a little bit too much. So we just didn't do that because we thought we'll wait until we purify these compartments more cleanly to really go down the specific transcripts that are localized to the ER.
All right, thank you.